This is part 1 of 2 on Chapter 8, looking at microbial metabolism. So in Chapter 7, we looked at nutrition and a little bit about the different types of things that organisms need in order to survive. So this is the actual metabolism. We're going to look at specifically cellular respiration and photosynthesis. So metabolism is just all the chemical reactions and physical reactions or workings inside of a cell. And if you look on the diagram on the right, if we start at the very top on the left, so sources of energy, organisms get energy from carbohydrates, lipids, or protein. These sources of energy, so the food or the nutrients, they go through a catabolic reaction called cellular respiration, and this releases energy in the form of ATP, which is in the middle of the diagram. And as byproducts of this catabolic cellular respiration process, we get carbon dioxide and water that's released. Okay, so this ATP, this energy that we get out of our catabolic reaction, we then can use that energy to do anabolic reactions where we take the building blocks of our different biological molecules, so like the sugars, the amino acids, the nucleotides, and the fatty acids, so those really simple building blocks. We take that ATP energy and you do anabolic reactions, and basically you're just putting those building blocks together to make the macromolecules, so those carbohydrates, lipids, and proteins. And then those macromolecules can be used to build up cells or to reproduce. So this is kind of an overview of all the metabolism that's occurring in these cells. These metabolic reactions, they are kind of slow, so usually chemical reactions are kind of slow if they just have to happen on their own. So in biology, all these chemical reactions, each reaction has something called an enzyme. So these enzymes, they're biological catalysts, which just means that they increase the rate of these reactions. It helps to speed up these reactions. And it does that by lowering something called the activation energy, or energy of activation. So the energy of activation is the amount of energy needed to get a reaction started. So in order to start any reaction, we have to put a little bit of energy in it. You have to give it a little bit of a push to get things going. And if you look at the graph up here, so across the bottom on the x-axis you have the progress of the reaction. So on the left you have your reactants, your starting products, and the reaction goes, and then on the right you have your products that are formed out of the reaction that's happening. On the y-axis we have our energy state. Okay, so if you look at the the dark green line, so the dark green line, it represents the amount of energy, so you have to climb up this energy hill in order to get the reaction started. And with the dark green line, that's how much energy you need without an enzyme present. So you need a lot of energy if you don't have this enzyme available. Whereas if you look at the light green line, that's the amount of energy or the hill that needs to be climbed with the enzyme present. So enzymes, they just lower how much energy is needed to get things started. And that allows things to go faster because you don't have to input as much work into all these reactions to get them going. So one important thing about enzymes, they're really, really cool molecules. But one thing is that the enzymes are not altered in the reaction, so enzymes can constantly be reused. So it's very efficient. Once they do a reaction, they can be used right away again to do another reaction. So they're very good molecules. So enzyme structure, so these enzymes, these biological catalysts, some enzymes are just really simple. They're just a protein that can work alone. There's other enzymes, so oleoenzymes, which are made up of a protein portion, the Apoenzyme, which is a protein, and then there's a non-protein portion called a cofactor. So this is where we have, most of it's made of a protein, but then you also have this other molecule that's kind of the helper. Some of these cofactors are metallic cofactors, so you might need iron, copper, magnesium, so some type of metal that will help with this reaction. Other cofactors are coenzymes. These are organic molecules, so molecules that are carbon-based. They have a carbon-hydrogen bond in them. These are usually vitamins, so like vitamin C, vitamin A, so that's why you need to, eat food with lots of vitamins, so you have these coenzymes available so your enzymes can work and your cells can do all the metabolic reactions. So that's why vitamins are really, really important. So this picture is showing different types of these enzymes. So the protein part is in purple, and then you can see these different enzymes. They can have a metabolic cofactor on the left, a coenzyme. They can have a coenzyme and a metabolic cofactor. So they can have one of these cofactors, or they can have multiple cofactors that help with this enzyme. So these enzymes are the protein part of your enzymes. Proteins, if you go back and review on when we talked about organic chemistry, I think it was chapter 2. So we talked about proteins, and proteins are structures or amino acids that are hooked together. So you take these amino acids, these building blocks, you hook amino acids together. That sequence of amino acids is called the primary structure. Then those amino acids start to interact with each other. They start folding, so you get secondary structure. So if you go back, you have alpha helices and beta pleated sheets. And then those alpha helices and the beta pleated sheets interact with each other and form bonds, so you get a tertiary structure, the third level. If you have multiple polypeptides that work together, so we have a purple one and a pinkish one. So if those join together, then you have the quaternary structure, which is where you have multiple polypeptides forming your final protein. Alright, so how enzymes work. So we have enzymes which are made up of proteins. So enzymes, they have a region on the enzyme called the activation site, or the active site. This is the region that actually does the reaction, does a catalytic or an anabolic reaction. So what happens is your substrate, which is your reactant that you're working with in the reaction, that substrate binds into the active site, and you get what's called an induced fit. So the temporary enzyme substrate union, it causes the substrate to go into this active site, and then the protein kind of hugs that substrate. So it's an induced fit. And then one other thing about proteins is they're very specific. You can see up here. So in diagram A, we have two different substrates. One of them fits, one does not. So enzymes are very specific to what type of reaction they're going to do. And then figure D at the bottom, that's showing that induced fit. So your substrate goes into the active site on the enzyme, and then the enzyme's going to kind of hug that substrate to keep it in place. Right, and then the cofactor part, I kind of talked about these already, but cofactors, they're micronutrients. So remember, those are nutrients that we need, or organisms need, but they only need a little bit of it. So these micronutrients, the vitamins and the minerals, can be used as cofactors. And these cofactors, they just assist the enzyme in its activity. So they just help to form bonds, or they help for the substrate to fit into the active site, or they just help to lower that activation energy. Enzymes can be located in different areas of the cell. So if we look at an exoenzyme, these are enzymes that are created inside of the cell, but then they're transported extracellular, so they're transported out of the cell. And these exoenzymes, their job is usually to break down larger food molecules or harmful chemicals. So they break down food molecules, and then the cell can take in those smaller food molecules. Or if the exoenzyme breaks down chemicals, they break it down before it gets into the cell. So different types of exoenzymes, we have cellulase, that breaks down cellulose. Cellulose is a very large carbohydrate. So cellulase breaks down the cellulose, and then the cell can ingest the smaller pieces. amylase, penicillase, that enzyme breaks down penicillin. So we use penicillin to treat, well, we used to use penicillin to treat a lot of bacterial infections. But now bacteria, they have this enzyme that they create called penicillase. Penicillase actually breaks down penicillin before it gets to the cell. So it's kind of a defense mechanism for these cells. So they're destroying the antibiotic before it even reaches that bacteria cell. Other types of enzymes are called endoenzymes. These are enzymes that are just retained intracellularly, so they stay inside of the cell and they function there. Most enzymes that cells make are endoenzymes, so they're used for the internal metabolic reactions. We can also categorize enzymes based on when they're available. So constitutive enzymes, they're always present, always produced in equal amounts or equal rates, regardless of the amount of substrate that's available for those enzymes to work on. So those are always there. On the other hand, you can have enzymes that are regulated. So these enzymes are not constantly present. We can actually turn off their production or turn it on. So if you turn on production of an enzyme, you induce it. If you turn off the production of an enzyme, it's repressed. And turning these on and off, we'll get into how this actually occurs later on, so how to turn on and off different genes. But this is just a response to the environment, so substrate concentration. Some enzymes do a synthesis reaction where they take two things and put them together, so that's synthesizing, putting things together. Whereas other enzymes do hydrolysis reactions where they're going to break things apart. So enzymes can do lots of different things, putting things together or breaking them apart. So enzymes, I mentioned that enzymes can be used over and over again, so they're not used up in reactions. Enzymes also operate under specific temperature, pH, and osmotic pressure environments. And those enzymes, you'll see a correlation between enzymes work best at a certain temperature and the organism grows best and reproduces the best at that same temperature. So enzymes and organisms' growth and development, their environments go together really well. So when enzymes are subjected to changes in the organism's habitat, they can become unstable. So these chemically unstable enzymes can occur when the temperature goes out of the optimum range, the pH goes out of the optimum range, or the osmotic pressure goes out of that optimum range. So this is kind of going back to chapter 7, where we looked at the different temperatures that organisms can grow at, the different pHs, the different osmotic pressures. So enzymes kind of have that same curve, like the temperature curves that you saw in the previous chapter. So these enzymes, if they denature, the bonds in the enzyme are actually broken. And if you break those bonds in the enzyme can no longer work. This really happens when you get to really high temperatures. The enzymes are going to denature, they're going to stop working. So that's another really interesting thing about enzymes, is they're very specific to what environments they work best in. Okay, so these enzymes, you find enzymes in things called metabolic pathways. So metabolic pathways are reactions that have multiple steps to them. So if you look in the top left, just look at the linear metabolic pathway. This is where we take molecule A, it goes through reaction to turn into molecule B. B goes through a reaction and turn into C. C goes into a reaction D and E, and so on. So this linear reaction, it has 1, 2, 3, 4 steps to it, and it can be even longer than that. So each one of these steps has a specific enzyme that's going to help speed up that reaction. So this linear system right here, since it has 4 steps to it, you're going to have 4 different enzymes, one at each of those steps. So you can have these linear metabolic pathways. You can also have cyclic pathways where molecules come into the cycle, and then molecules will exit out of the cycle, and you always get back to your starting molecule. So we're going to see some of these cyclic pathways in a little bit. Other types of pathways, you can have divergent pathways where you start with one product and then it can go down two different pathways, or convergent where you have two different reactants and they converge together into one pathway. Most of the pathways we're going to be looking at are either going to be linear or the cyclic ones, but do be aware that there's divergent and convergent ones as well. Enzymes can actually be controlled, so there's two different direct controls on the actions of enzymes. So there's something called competitive inhibition. This is where you have an enzyme, shown in purple, and then you have the normal substrate the enzyme works on, and then you have something called a competitive inhibitor that has a similar shape to the substrate. So in competitive inhibition, this normal substrate and the competitive inhibitor, they compete for the active site. If the normal substrate gets into the active site, so the dark brown substrate gets into the active site of the enzyme, then the reaction is going to proceed, which is shown going to the left. If the inhibitor gets into the enzyme active site, that reaction is going to be blocked. You're going to inhibit that reaction. So competitive inhibition, you have this competitive inhibitor that resembles a normal substrate, and they compete with each other to gain that spot. So the more competitive inhibitor you have, the more chance you're going to turn that enzyme off, so you're not going to have the normal reaction. So it's a competitive inhibition, it kind of slows down how enzymes work. Non-competitive inhibition, this is more of a way to actually turn off an enzyme. So this is where enzymes are regulated by molecules that bind to an area that's different than the active site. So normally we have our substrate up at the top going into the active site of the enzyme. You go to the left and the reaction proceeds to produce the product right here. That product can go back and bind into something called the regulatory site, and that's going to cause the active site to change shape so your substrate can no longer bind to it. So non-competitive inhibition, we're actually turning that enzyme off so it no longer works. The creation of enzymes can be turned off at different parts during protein synthesis, so we can actually turn off genes or turn off the DNA so we don't get the protein created. We can have those different types of inhibition occur so the protein doesn't work, the enzyme doesn't work at all. So I'm not going to go into a lot of detail on this slide, but just know that enzymes, we can turn them off in multiple ways.